Well, welcome to the second video on the key area 2.6, and we're going to be looking at patterns of growth in microorganisms. Now, the first thing we need to do is actually define what we mean by growth. And I've got written here, growth is when the rate of synthesis of new material exceeds the rate of breakdown of material. So what we're seeing here is if there is an increase in bone in humans or an increase in muscle, that results or exceeds the breakdown of that results in growth. Now, there's this line here that says it involves an irreversible increase in dry biomass. What we're talking about here when we're speaking about dry biomass is removing all the water from an organism. Because if you were going to be weighing something to see if it's grown, then it could be that there's water that's in there. It's not really a very reliable indicator if you've got water present, because that will vary from day to day, if it's a plant or if it's an animal. So what you want to do, if you want to see if growth is happening, then the best way to do that, or the more reliable way, is to look at the biomass, the dry biomass that's been produced. When it comes to bacteria and yeast cell growth, we can measure cell growth by looking at the cell numbers that are being produced over a period of time. The cell numbers can be counted, and that can give us an idea of how much growth is happening there. So when we look at a microorganism dividing, the time taken for a cell divide is called the generation or the doubling time of that cell. So we have a wee look at microorganisms and the kind of pattern of growth that happens with them. This is a typical graph showing the growth of a microorganism, and there's a number of different phases that are there. The first one we're going to have a look at is this one here. It's called the lag phase. And during the lag phase, there's little or very little increase in cell numbers. That you could think of it in terms of, if you imagine the cells are growing in a fermenter, and what's happening is they're in there, you've added the nutrients, the media that we've been talking about in the last video, we've got all the pH right and all the oxygen right and everything like that, and the cells are starting to get used to these conditions and they're starting to begin to metabolize. The next stage we've got here is the log or the exponential phase, and what is happening here is that the cells are starting to grow and starting to multiply at the maximum rate, provided that there's no limiting factor that's there. So we're talking here, it's called the log phase, because we're talking here about where you've got one cell, it will divide into two cells, and then each of these two cells will divide again, and then each of these two will divide and produce cells. And so what happens is you get this doubling of numbers as you're going along. This is called the exponential growth, because you're going from one cell to two cells to four cells, and then you're going to 8 cells, 1, 2, 3, 4, 5, 6, 7, 8, yes, and then you go to 16 cells and so on. And it doesn't take a long time as the numbers are doubling for you to end up with a lot of microorganisms there. The next stage we've got is the stationary phase here. And what is happening here is the nutrients are beginning to run out. Some waste metabolites are starting to build up. The waste might be toxic to the microorganism. It might slow down the rate of division. And eventually what happens is that you start to get this death phase, where there's lack of nutrients, accumulation of toxic metabolites, cells are dying and no new cells are being produced. So the population is declining, and it might disappear or it might survive just as a few resistant spores. Now I want to have a wee look at this log or the exponential phase of growth, where the cells grow and multiply at the maximum rate as long as there's no limiting factors. And if we were to plot that on a graph, we would get a curve here that looks something like this, where we're looking here at the time down here and the cell numbers here, just in an ordinary arithmetic scale. And you'll see here we get this quite tight curve here, and then it curves up rapidly like this. The thing is that using this type of graph can be a bit of a problem because our cell numbers are doubling every time, and you can end up in this phase of growth with a cell number that's really, really large after a few hours, which makes it pretty hard down here, for example, to be really plotting accurately. If you're just looking at a cell that's dividing every half hour, you know, at the point of 6 hours, you've got something like 4,096 cells. So it makes the scale here kind of pretty tricky to be working with. And there are a number of ways that we can get round that. So I've just talked about how tricky it is to be plotting this graph of growth on a sheet of normal graph paper. And one way of solving that problem is to use a scale that doubles in number each time going up the y-axis. So here, when you look at this graph here, you'll see that each of these is plotted and the numbers are doubling. And then if we've got the situation where we plot, we've got one cell to start off with, and then if we're talking about a cell doubling every half hour, we've now got 2 here, and then when we reach the 1 hour mark, we've got 4. When we reach the 1.5 hour mark, we've got 8. When we reach the 2 hour mark, we've got 16. And when we plot a graph like that, then what happens is we get the growth as a slope, as a straight line, or equal slope, a line of equal slope. Now, as if graph drawing wasn't complicated enough, there's this other type of graph drawing that can be done for this log phase. And that's using Semilog paper. It's called Semilog because one side of it is just an ordinary linear scale that you would be used to with graph paper. So here it's the time. At this side here, though, the cell number, the axis that is here has got these cycles here. So this is a cycle, here's another cycle, here's another, there's 4 of them. Oops, that one's not drawn very well, that's going right down to there. And you'll notice that the lines on the scale are getting closer together as you go up each cycle, and each cycle is made up of 9. And what we're looking at here is this scale. If I just draw out what the actual axis is talking about, I'll just rub this out so we can, and I'll plot out. There's no log of 0, so we start with 1, and that is down here. So this is 1. Here is 2, 3, 4, 5, 6, 7, 8, 9, and up to 10 here. Now, what happens is that in each of these successive cycles, the values are 10 times greater than the cycle before. So when we get to here, at this point here at 10, now we're going up to 20, 30, 40, 50, and so on up till we get to 100. And then in the next cycle, it's now 10 times greater again. So this here will be 200, 300, 400, and so on up to 1,000. And then the next cycle here, that will be 2,000, 3,000, and so on. And so if we're looking at plotting a graph where again we're looking at cell numbers doubling every half hour, so we've got 1 at 0 time, then we've got 2 cells, then we've got 4, 8, 16, 32, 64, and so on up till we get to 4,096 at the 6 hour mark. What we'll be doing is we plot, first of all, our first plot at 0 hours, here is 1. Then half an hour later it's 2, half an hour later it's 4, half an hour later it's 8, and then at the 2 hour marks, well, it's 16, so that is in here somewhere here, just past the halfway mark. Then another half hour later, we're at 32. So here's 20, here's 30, so 32 about here. And so on. Then we come up here, up to 64. And then we keep on plotting using this scale till we get to the 6 hour mark, where 4096 is up here. And that is how to plot a Semilog graph. There are other types of graph paper where both sides are logged, but fortunately you only need to know about the one. Page 197 and page 198 in your textbook have got these graphs in them, so you can have a look at them there as well. So you've survived the graph drawing part now. Now we're going to just have a wee brief look at some of the controlling metabolism items that are contained within this key area. And one of the things you need to know about is this idea here, where some microorganisms, in the example they've given are filamentous fungi or sometimes bacteria, they've got two different types of metabolism. There's this primary metabolism, and that occurs during the active part of growth, or in other words, the lag phase that we've just drawn all these horrible graphs for. What's happening there is the microbes breaking down substrates that are there that have been given to the organism that we've got in a fermenter that we're wanting to grow, and we're giving it amino acids, we're giving it all sorts of other things for growth, and that's the primary metabolism that's occurring. Now the other type of metabolism that occurs is at the very end of this exponential phase or during the stationary phase, and what happens there is that the organism's been doing a whole lot of division, and they now produce secondary metabolites. Now they may be used for cell, they maybe not used, for cell growth or cell division, but they might have some sort of ecological advantage for the fungus that makes them. So for example, things like penicillin that are produced by certain fungi, well, that confers on them some sort of advantage because they can produce this antibiotic, so it stops bacteria growing round about them and it helps them to survive. And that's what we mean when we're talking about secondary metabolites or secondary metabolism. I just mentioned on the last slide that secondary metabolites, an example of that would be an antibiotic which inhibit microbes, for example bacteria. So if you've got fungi in the soil, then that helps to protect the fungi from bacteria invading them. But there are other secondary metabolites which we grow the organisms because they produce useful products. And there are certain yellow pigments that can be produced, or analgesics, that is painkillers, or antibiotics that I've mentioned, or certain immunosuppressants or drugs that are used in ulcer treatment. All these are secondary metabolites. They're produced by the organism to give the organism some sort of advantage. They're obviously not producing that metabolite with the view in mind that we would produce tablets from it in order to treat human diseases, but they're there for that organism to help it have some kind of advantage in the situation that it's in. But we make use of this by growing these organisms in these large fermenters and then packaging them up, putting them into bottles and selling them at exorbitant prices in the pharmacy. Now imagine you've now got your microorganism, it's growing away. You've done tests to have a look to make sure that it's growing well, and during that log phase, and it's growing well during that exponential growth, and you've drawn all these graphs to show that and so on, and you've got your industrial fermenter and you've got your organism there, and it follows this pathway. It's going from some sort of metabolic substrate A, it gets broken down by enzymes to B, and then more enzymes to C, and more enzymes to D. But the thing is that you want this desired product here. You're naturally not really all that interested in producing lots of metabolite D. it's this here that you're wanting to produce. So what can you do in order to make sure you produce lots and lots of this? Because obviously you are an industrialist and you've got this business and you've got these microorganisms growing in the fermenter, and you don't want them to be producing lots of stuff that you don't want. So let's have a look at the ways that we can control this. So here you are, you're a wee industrialist and you're producing this very useful metabolite here. And you're wanting to make sure that you produce that and not this here, because that's just going to be wasting your money. So what can you do? Well, you can continuously provide this metabolite here, metabolite A. And that's, when we do that, we use that term, we call it a precursor, in order for it to be changed into B, and then in turn that will then get changed into this metabolite here. And an example of that is something called phenylacetic acid, and it's added as a precursor to help in the production of penicillin. Or the other thing that you could do is you could provide lots of metabolite B, and that acts as an inducer. So you're adding that metabolite, and what happens is the enzyme will then break that down to the substance here, C. As well as that, what you can also do is supply an inhibitor here to enzyme 3 so that this part here of the pathway doesn't occur. So you build up lots and lots and lots of this substance here, and that hopefully will then make you lots and lots of money. And that is the end of this key area 2.6.